Our next speaker is Patrick Horn. Patrick is leading the clinical development at Orphan Technologies. Prior to joining Orphan Technologies, he was a pediatrician in the Chicagoland area for 20 years, and then he moved into drug development leadership positions. He worked for Abbott Laboratories, Diex, and Tetraphase Pharmaceuticals. And then within, I think, the past year, you can correct me if I'm wrong, he moved over to Orphan Technologies and is working on the enzyme replacement therapy for HCU. So I'll let you take it from here. The wireless microphone is also on if you prefer not to stand.
And thank you, Danae, and really thank you to the HCU Network America. I think it is an incredible experience and an incredible honor to be here. I think Ben yesterday pointed out that this was the inaugural meeting, and I think that you guys should be very proud of what you've done so far and very excited. And I think, because I think you're going to find that you're going to get incredible power with this organization. I think that Dr. Levy pointed out the individual benefits, the fact that you have a sense of community, that you can teach each other. And yesterday we heard about individuals finding their own voice and being their own advocate. But this association can really be the voice for a community. And I think that you will see, as you start to use that voice, it's very powerful. Use that voice. Get in front of companies that are doing clinical trials. Really make sure those clinical trials are designed to answer your questions. I mean, we have a lot of regulatory questions we have to answer, but make sure you know that you get what you want out of them as well. Get in front of the diagnostic companies. Tell them you want a finger stick HCU, that this is really what you need to really manage it effectively. And really, I think you're going to find how incredibly powerful that is. So again, that's a very long-winded thank you, but really thank you for having me and Orphan Technology to be a part of this. I think so my task today is talk about enzyme replacement therapy and talk about the upcoming trial that Orphan Technology is sponsoring. I think one of the advantages of being kind of late in the program is that a lot of my initial slides have been presented and have been kind of discussed and have been presented by people much more knowledgeable about homocystinuria than I am. But I'm still I'll go through them, I'll try to pull them together and to kind of let you know how we're thinking about it and kind of where we're going from here. Okay, and so for disclosure, obviously I am a full-time employee for Orphan Technology. So conflicted, but that's where I am. You can't talk about homocystinuria without talking about protein. And protein is an aspect of every part of homocystinuria. So about 40 to 60% of the human body is made of proteins. And this varies from structural proteins, your bones, muscles, your hair. It's the enzymes that perform millions and millions of different tasks within the body. You know, we're here focused on one specific enzyme, CBS, because that's the one that's not working quite right for us. But it really is just amazing how many there are. They're transport proteins that carry compounds and chemicals from one part of the body to another. They're the immune response, the antibodies, that people, the way we fight viruses. They're all proteins, and they're cell signals. Excuse me, I think yesterday Linda pointed out that proteins are made of amino acids, and there are about 20 different amino acids. And she had the picture and called them kind of a string of pearls. And I think that's a beautiful way to kind of think about how proteins are built. They're built from individual amino acids. There are countless proteins in the body, and they're all built from the same 20 amino acids. But the way those amino acids are strung together, the way they fold after they come back, gives you everything you need. 9 of these amino acids we've heard are essential amino acids, meaning the body can't make them, so we won't go into that. But the other thing to remember is proteins are continuously being built and continuously being degraded. The body has this incredible system where it kind of maintains a constant. And so people are building up proteins, people are taking down proteins. So at the same time your body is incorporating these amino acids into proteins, somewhere else in the body these proteins are being destroyed to release amino acids. And that becomes all very important in terms of when you try to modify it. That also means that your protein requirement varies considerably across the lifespan and from just day to day. So for example, a child during the growth period, it's a net gain of protein in the body. You know, your child grows, it gets muscles, it gets bones, he or she continues to develop. So it's a net gain of protein. So their protein requirements are going to be larger than an adult's. And as an adult, you may think your protein requirement is stable. And for the most part, it's more stable than a child, but that can vary from day to day. Whether or not you have a cold is going to impact how much protein you have. You know, clearly pregnancy, when you're building a whole other body, impacts your protein. You know, if you start to go to the gym and you get fit and toned and you get your six-pack, your protein, you go up, you build muscles, all of that. So that's why when you go to the doctor sometimes, although you've been following your diet, you've been very good, your homocysteine level's not quite on target. Converse of that is you go to the doctor, you're kind of dreading because you know you haven't been quite as good with your diet, but your homocysteine level's right on target. So it's all just part of this kind of flux. Then when you get to be kind of the other part of the age, like my age, you kind of have a net protein loss. You know, I'm starting to lose, at my age, I lose muscle mass, I lose bone. I've lost an incredible amount of protein just in hair loss. So it's really changes your day-to-day protein requirement. So I think my talk is probably the last one where you're actually going to see this cycle. And you'll probably see this cycle tonight when you close your eyes. I actually have it on 4 different slides because it really helps to talk about it. And it is important. It's exactly the same one that everyone is presented, the methionine, the homocysteine, the cystathionine, and the cysteine, and then the other pathways. So really, again, methionine is an essential amino acid. That's been said before, but that's important because that really is the reason that dietary methionine restriction can work and can be beneficial. Because if your body could make methionine, as Dr. Levy said, you know, it wouldn't matter how much you did. It's normally metabolized to homocysteine, cysteine, and is assisted by B6. And in homocystinuria, there's a lack of the CBS, which is down there in the green. And then a buildup of homocysteine in the circulation is a hallmark of the disease. Actually, a buildup of homocysteine in the urine is how it was identified. And the way this works is, so this reaction, this cycle goes on inside cells. So inside the cell is the first place the homocysteine builds up, and then it spills out into the blood, and then it spills out into the urine. So it's kind of like a blocked sink. And so you get your buildup of homocysteine. You not only get a buildup of homocysteine, but everything in front of that block gets built up. So you get an elevation of homocysteine, you get an elevation of methionine, and everything below that block gets decreased. So cystathionine and cysteine get decreased. As Dr. Levy says, the focus of all of the research and kind of the weight of the evidence really kind of indicates that it's homocysteine that causes the impact. And that's why everything is really targeted at controlling the homocysteine levels. And kind of here we talk about it. So, you know, your B vitamin therapy. Vitamin B6 obviously really is, we've talked about that, we've heard about that. It's a cofactor. It can really drive kind of the degradation of homocysteine to cystathionine. Vitamin B12, some of you may be on B12. So if you look at that, that's on the left-hand side. So that kind of is important in maybe helping drive the conversion of homocysteine to methionine. And the folate too, for those of you on folate. that's where it all kind of fits in. I think yesterday you raised the question about, converting homocysteine to methionine doesn't really reduce the total amount. But that's because the attempt is to reduce homocysteine, and you're right, by converting it to methionine, some of that methionine, especially if you're actively building protein, may be taken up to a new protein, but some of it's just going to cycle back down to homocysteine. But the important thing that all of these impacts are trying to do is really trying to control homocysteine. Betaine is the same way. Betaine converts homocysteine to methionine. Increases your methionine level, yes, but decreases your homocysteine level. And again, and this is because, the focus on therapy is really decreasing homocysteine levels. Are these other elevations of methionine or decreases in cystathionine and cysteine important? Dr. Levy said, there's some ongoing work. There's maybe some hints that there are. It starts to get very complicated, but it really seems that it's the homocysteine. So given all of this, there really is kind of this unmet need for novel therapies, and novel therapies to further decrease homocysteine and to maybe decrease homocysteine and still allow some dietary relaxation. So this is where then the OT58. So OT58 is an enzyme replacement, and it's not possible right now with current therapy to actually cure the disease. And as Margie says, as other people have talked about, gene therapy, gene editing, this CRISPR technology, I'm certainly no expert on it, but CRISPR technology I think is an incredibly powerful and exciting thing. So maybe a cure for homocysteine is on the horizon. And right now, it's just not there yet. So what we're talking about today is enzyme replacement therapy. So enzyme replacement therapy is not going to cure the disease, but if it proves to be effective, could be a powerful treatment. And what it should do, if it's effective, is normalize the level of not only homocysteine, but of all the metabolites. So it should normalize the levels of homocysteine, methionine, cystathionine, thionine, and cysteine. As Margie said, OT58 was developed at the University of Colorado initially, and then it was kind of licensed in by Orphan Technology, and Orphan Technology has really kind of been the driver behind the fine-tuning and the duration. And a little later in the talk, we'll talk about kind of the steps in drug developments and how it is. But, for OT58, so initially it was a recombinant human enzyme. The initial trials were IV, and some were given sub-Q, and we found that, yes, it worked, it lowered it, but it didn't last long enough. So the modifications were made in terms of how it's delivered, how it's given. It's now a product that is given, presumably once a week, but as Margie says, we've never given it to humans before. But really, it really has been very effective in the animal models, and I had a little bit of data on animal models here. So this graph, kind of looks like a squiggle, like your child may draw. But what it is on the left-hand side are the metabolite levels that you look at, and there are 3 of them there. So the orangey red one is homocysteine, the blue one on the bottom is cystathionine, and the green one is cysteine. And on the bottom then is plotted days, and this is a study in mice. I should have said that earlier. And so where there's a gold bar on the bottom are the days when the mice got injected with OT58, and when there's no gold bar, there was no injection of OT58. So you see there are 3 different periods of treatment with OT58 in the mouse model. And what this does, look first at the orange line, and you can see during that first period, the levels of homocysteine drop from above 150 down to about 25. And they drop down. Because this was an early study, as before the enzyme was modified, it was a relatively quick go back up. It went back up to about the same pre-treatment level. The second treatment came in, and again it dropped. And then that was stopped, it went back up, and then there was a third treatment. It came down. At the same time, then the downstream metabolites, the ones that are after the block of the CBS enzyme, actually show the opposite response. So if you look at cystathionine and cysteine, they actually go up when homocysteine is coming down. And that's exactly what you would expect to see, and the results you would expect to see if, in fact, you were replacing that enzyme activity. So, this very early mouse model said yes. So this could in fact work. So, again, there's been a long progression, and really then what we look at is it reduces homocysteine levels in the plasma. And earlier I said that, that cycle that's now burned into everyone's brain takes place in the cells. So the question is, okay, we can reduce homocysteine levels in the plasma, does that mean we're reducing them in the places where it really matters? And in the mice, it does. When you reduce homocysteine levels in the plasma, it reduces it in the tissue, and that's because homocysteine can easily cross cell membrane. So you're metabolizing homocysteine in the plasma. It leaks out of the cells. You metabolize that homocysteine so that if you look at the liver, if you look at the brain, if you look at the heart, if you look at other tissues in mice, you can normalize homocysteine in those tissues by decreasing the plasma homocysteine levels as well. There are a number of different mouse models of homocysteine that have been genetically engineered to either have a mild form of the disease or a more severe form of the disease. If you look at those mouse models, you see a number of effects, and you can reverse those. So in the most severe mouse model that have just severe homocystinuria, no enzyme activity at all, then those mice pups die at a young age, between 20 and 30, and die predominantly from liver disease. OT58 can prevent those deaths. It can act as an enzyme replacement. It can prevent those deaths in those mice. And if you look at the other mouse model that's less severe, it can prevent the complications of homocystinuria. It can prevent the development of progression of osteoporosis and the other ones as well. So the mice data really clearly then are very exciting. And not to just have slides without pictures, I put a couple pictures here. And so that top, on the left-hand side, you have healthy mice. And then in the middle, you have mice with homocystinuria. And that HCU 1278T is just the type of mouse model it is. And this obviously is a mouse model that is less severe because they grow up to adulthood. And then on the right-hand side are mice that are treated. So that one where you have the pictures of the mice, you can see that in the healthy mice, they have a normal mouse face. But interestingly enough, in this mouse model of homocystinuria, in the middle one, with this mouse model, they kind of lose the hair on their face. And it's called facial alopecia. And there's nothing similarly equivalent in humans in terms of that, but in the mouse model, it's there. It's related to homocystinuria. And the fact is that when you treat it with OT58, they regrow the hairs on the face, or it prevents the loss of hairs on their face. So clearly that mouse effect is there. The other thing is then if you look at the eye, which is related to humans and how it goes, is you can see on the left-hand side is the normal ciliary zonules, and in the middle there is the affected mouse. You can see the gaps, you can see the breaks. And this is, when enough breaks happen, this is when you see lens dislocation. And then you can see mice treated with the OT58 really have normal eyes. So it really kind of demonstrates that in the mouse model that we can treat homocystinuria. We can treat, and by enzyme replacement, treat that. And I think that's key. The mouse studies are all very exciting, and they really give us what's called proof of concept. It really starts to demonstrate some efficacy. But again, I think everyone needs to remember that these are mice, that these are mice. The models look good. The models have been validated. We're very excited about the results. but these are mice. We have not yet given OT58 into humans yet, and that's where the first study is. And so we have, what do we do? What is the next step in a drug development pathway? And this is where, to the uninitiated or even the initiated, it seems like an incredibly long, complex process. So what we need to do in terms of having a drug that we can take into humans to do our first trial and ultimately to make commercially available is we have to have a manufacturing process where we know exactly what we're making, and every batch has to be exactly the same. We need to have safety. And we start out, we look at, we'll talk a little bit about it, we look at safety in animals first, and then we look at some small studies in humans, and then we expand, and we go. We look at efficacy, and this is where in these genetic diseases, what do we need to do to demonstrate efficacy? You know, for us, it seems very reasonable that just correcting the metabolic parameters by reducing homocysteine, by reducing methionine should be enough. FDA has a little bit different view. They need to show that not only we're affecting the biochemical marks, we have to show a clinical response. We have to show that impact by demonstrating, by reducing these biochemical markers, excuse me, and keeping them low, excuse me, we can actually demonstrate a clinical response. And that's where the natural history study is very important, because we need to know over the span of a 3-year time, which is kind of what we think on the range our pivotal study will be, what can we expect from a patient who's on current therapy, and where we're going with that, and what are the results going to be. And even the biochemical response, you know, people have been studying homocystinuria for a long time, but what is the day-to-day variation of a homocysteine level? You know, is it 20%? Is it 30%? What kind of reduction do we need to have to really demonstrate that this is due to the drug and not due to the fact that someone snuck a hamburger or someone was really good and didn't sneak a hamburger? So all of these are kind of things that are going on. I think I may have missed one. So again, then when we go to move on, the things we really need to do are manufacturing. And this is an incredibly complex problem, especially when you're talking about a biologic. It's like, again, we need to demonstrate that every lot, every batch of OT58 we produce is the same, that it has the same composition, that it's going to act the same. And the reason for that is, you know, we'll produce a batch and do our toxicity studies in mice. We'll produce a batch and we'll do our toxicity studies in monkeys. And we'll demonstrate, you know, that it is overall safe in a mouse. But then we produce a batch for humans. So we have to demonstrate that that is exactly the same drug as we studied in the mice and the monkeys. We have to show, again, that we can, it's one thing to make these in a lab at a university setting, to make a very small amount, but how can we make enough to really supply a clinical study? How can we make enough that will really supply once we get on the commercial scale? How do we measure this? The analytic test. One of the things we have to do is demonstrate how much of this OT58 is in the blood, how much is circulating. How do we do that? Can we have a test that really gives us a definitive answer about how much of that? And the formulation, you know, I said we kind of tweaked the formulation a little bit to make it kind of what we think will be a once-a-week injection. Can we do better? Can we make it so that there's a smaller volume that we inject? Can we do it so that it's something that right now it's stored in frozen conditions? Can we make this room temperature? Can we tweak these formulations? All of that's going on, and stability as well. In terms of what we need to do to demonstrate safety and efficacy, so then, you know, the steps of this are, there's the preclinical, the animal studies, then there's the clinical studies, and there's probably also then going to be some post-approval studies, you know, whether that's a registry or what. we have to demonstrate efficacy. We start out in animals, and then we go into humans in the early clinical trials, and I have a slide to kind of look at that, and post-approval studies as well. So this is kind of the development pathway, and we start out. And this is kind of the red arrow is where OT58 is, and it's kind of what we've done. So we've done the proof of concept. We've kind of shown that it works in animals. We've done the animal pharmacology, so we know when we give a dose to animals kind of what it does and where it goes and stuff. We've done the toxicology studies. For every one of these steps, the FDA has very specific requirements of what it is. So you have to do a toxicity study in 2 species of animals. Usually one is a rodent, and usually one is a non-human primate. So we've done those studies. Then after you get that, you have your initial manufacturing. You have to demonstrate your stability. You have to do all of that. Then after you get to that step, you put all of that information together, and then you present it to the FDA or the EMA. You know, right now we've gone to the FDA. We haven't gone to the EMA is the equivalent of the FDA in Europe. We have an automatic build apparently. And so that's where we are. You know, the FDA has reviewed the data. The FDA has said, yes, your drug looks safe, and there's a hint of efficacy here. You're authorized to do clinical studies. The other thing we did down at the very bottom of the light blue is we got an orphan drug designation. And you'll hear a lot about orphan drugs, and what that means is the FDA has some very specific requirements for orphan drugs. One of them is the population within the United States is less than 200,000. And what that does then is kind of change the regulatory pathway. And there are some differences with orphan drug development than with regular drug development. And one of the biggest ones is when you look at the right-hand side, the phase 3 studies, which are your safety and efficacy studies. So if we were developing a drug for hypertension or asthma with huge populations, we would have to do 2 separate pivotal phase 3 studies, and they would both have to be confirmatory. For an orphan drug, it's a single phase 3 study. So there are some differences in that as well. But again, so from a clinical point of view, what we've started is the natural history study. And again, that natural history study is going to start to address a lot of this. Like, what are we going to pick as the clinical endpoints for our pivotal study? What can we measure the most rigorously, the most consistently, and over the course of 3 years, we're going to expect a change. And so I think that is a critical piece as well. I think the other thing is that has been kind of talked about, and we've talked about it in terms of our survey. We've talked about, Margie mentioned it, in terms of kind of as a marketing approval, we have to demonstrate a health economic benefit from this. So we have to demonstrate, you know, that it is safe and it's efficacious. It really does what we say it's going to do, but we also have to demonstrate that it is cost effective, if you will. And so the natural history study will kind of will help us with kind of what is the economic and health burden of illness. Kind of the survey we were talking about initially, and that some of you have mentioned, is we're also going to have to start to tease out what is the humanistic burden of this illness, kind of what is the impact of this. So all of this is kind of coming together in the natural history study. I guess I'm talking longer than my slides think I should, but anyway. So where we are. So one of the other things about an orphan disease, so the upcoming study, instead of having a separate phase 1, then phase 2, then phase 3 program, and typically what that is phase 1 is done in the healthy volunteers. It's usually just to look at kind of pharmacokinetics, what are the drug levels, how are those done, and then you go into the phase 2, which is your proof of concept and dose ranging study, and then you go to your pivotal study. You know, we are doing a combined phase 1, phase 2 study as the initial study for enzyme replacement therapy. So we're not going into healthy individuals, we're going into a patient population, because that will allow us to do all of those things. We can do pharmacokinetics, and when I say pharmacokinetics, I mean we'll give an injection, we'll do periodic blood measurements pretty frequently over the first 24, 36 hours. We'll see how that comes up, we'll see how long it lasts, and that will help us determine dosing. And then we'll do it's a dose escalation study. So we'll start with a low dose. If we demonstrate safety and efficacy, we'll move up to another dose. We'll move up to the third dose. And the study will have up to 5 different doses and 5 different cohorts as we move along. And we've defined kind of what the doses we think will be for the first 3, because based on what we've seen in animals, we have done the models to kind of say, okay, we think this is the dose that's going to give us in humans the target, and this is the frequency we need to give it. But again, this will be our first in-human study, so maybe we don't need to give as much, maybe we need to give more, maybe we don't need to give it as often, or maybe we need to give it more often. So all of these will kind of be sorted out in this phase 1/2 study. In terms of a natural history study, Dr. Levy talked about it. Just on this, I think, there are 5 sites right now, 2 more coming up in the U.S. It's actively, since I made these slides, we have another patient, so we now have 34 patients enrolled in this study. And already the data we're starting to get from this. We're just now starting to pull out some of kind of the baseline characteristic data from this. And I had kind of thought maybe we'd have a little data to share for this meeting, but it's just too early yet. But we're trying to pull it out. We're trying to get it in front of some scientific meetings. But clearly next year, if we're fortunate enough to be invited back, or maybe Dr. Levy can present some of what we found in that, because it is going to be very interesting to really kind of pull out, what does the population as a whole look like? Where do people fall on this, and how are they doing with that? So with the first in-human study, so clearly, like I said, we've done our animal studies. Obviously, there are more animal studies that we'll have to do as we progress, but we've done enough that we can demonstrate safety to go into a human population. We've had the manufacturing. We have demonstrated to the FDA that we can consistently make this, and it's the same product every time. The FDA has reviewed our protocol for the first in-human study, and they have given that an okay. And they've given us permission to start. So we have started. We have approached the sites. So right now, we anticipate that this first study will have anywhere from 12 to 20 subjects, and it will just depend. So it is a small study, but for this first-in-man kind of dose-ranging study, it really, that's what we need. We think we'll have 4 sites. We'll have, and the sites we've identified so far are going to be Boston Children's, CHOP, Indiana University in Indianapolis, and University of Colorado in Denver. Pardon? Aurora, well, right outside Denver. Okay. And those are the sites. Obviously, if enrollment's slower, if we need to enroll even more cohorts to really fine-tune the dosing, we may expand the sites and stuff, but that will be our initial sites. I think, it is for patients who have an abnormality in CBS. So it doesn't matter whether it's pyridoxine responsive or non-responsive, but it does matter. What does matter is your homocysteine levels. You don't have to come off diet. You don't have to come off current therapy. In fact, you can't. You'll remain on your current diet and current therapy throughout the study, because otherwise we would never know what was changing your homocysteine level. If you come in on one diet and you change your diet, how's that going to impact it? So you stay on your current diet, you stay on your current therapy. In order to be enrolled, you need to have your baseline homocysteine level has to be 80 or above. And that's partly because right now we don't know what that fluctuation is going to be. You know, if someone comes in at 80, you know, the next time you draw their blood, is it going to be 60? And is that going to be a normal variation, or is that going to be due to the therapy? You know, even 50. So 80 is relatively high, and ultimately we hope, you know, clearly we would ultimately treat patients, and maybe even in the pivotal study be able to enroll patients. But right now, to get a sense of what we're doing and get to proof of concept, you know, then you need your patient of 80. And I think otherwise, I think the inclusion-exclusion criteria are pretty similar to regular studies and regular clinical studies, and there'll be a list of them. And certainly, you know, if you're interested, you can go to clinicaltrials.gov. Some of that information is on clinicaltrials.gov. You can stop by the table, and we can talk to you a little bit about that as well. But I think, you know, we're incredibly excited, and again, you know, with the advent of the patient association, with the kind of starting of the clinical trials. And again, I think you'll find that as you develop your voice, this won't be the only clinical trials. Margie outlined a number of other things that will be coming out in clinical trials. I think if companies see there is a vibrant community, they're really going to be much more interested and much more active. And also an entree into finding enrollment for the clinical studies, because in rare diseases that can be a big hurdle to development, is identifying your patient population. So, you know, if the HCU Network America can really say, okay, you know, we have the population, we're enthusiastic, we're really to look at and enroll in these clinical studies, I think you'll find that that's an incredibly helpful thing. I went too far this time. Okay, so that's it. But I'll certainly ask any questions. You can ask them here. We'll be at the table. Anything. I went too far.
Nope, that's okay. You just can go.
Okay.
There. All right. I had a feeling there was going to be lots of hands for this one. Okay. You can just pass it down.
Hi, I'm Karen Lewis. I have 3 children. 2 of them have homocystinuria. 2 of them are currently in the natural history study. My daughter's 16 and my son Benjamin is 26. My question is, when they say that the studies for 3 years that they're looking at their levels, do they wait the 3 years, or do you gonna start the enzyme replacement clinical trials earlier than the 3 years?
Okay, so the natural history study is separate from the clinical trial study. But patients in the natural history study will, as long as they meet their requirements, will be able to roll over. But if patients remain in the natural history study, that's what's called a non-interventional study. So if you stay in the natural history study, you won't get enzyme replacement therapy. In order to get enzyme replacement therapy, you'll need to roll over into the clinical trial.
And how does that process work?
Just talk to your PI.
Okay.
So PI will know. You know, I think that the inclusion criteria for the natural history study are very, very similar to the clinical trial. The couple differences are that the homocysteine level, the baseline homocysteine level, the other is the age. So in the clinical trial, in the enzyme replacement clinical trial, ultimately it's going to be ages 12 to 65. But you know, the FDA was pretty adamant that we had to show at least a biochemical improvement before we could enroll patients under 18. So the first cohort, maybe the first 2 cohorts, will be 18 to 65, and then if we show a biochemical improvement, we can enroll the younger kids. So the natural history study, I know, goes down to age 7, I believe, 5. So the youngest ones in the natural history study won't be eligible for this initial clinical trial.
And when are you expecting the initial clinical trial would start?
So we have started the process, you know, so once we heard from the FDA, we've done what's called the startup process. So we are in the process of identifying the sites. We've identified those sites. We've communicated that. Now we're putting together the package. and I think it's almost complete. We'll have the package to the investigators that they need to submit to their IRB. We'll have to do contract negotiation with this. Anyway, this is way more information than you want to know. The answer to your question is we hope to dose the first patient in the trial, the first cohort of patients in the trial, late summer, early fall of this year.
Okay, thank you.
That's our target.
Hi. Thank you, by the way, for all the information you've shared. This is certainly a complex process, and being able to convey it to us in a way we understand it, we certainly appreciate it. I think Margie mentioned this earlier about some other efforts. Can you speak to, I guess, your prediction on how this enzyme replacement might behave in the body? There's certainly been attempts in other metabolic disorders where they've attempted enzyme replacement, and there's been immune response, some rejection of the body because it is a protein. Is there anything that gives you confidence when you move into human that this will actually be effective, or is that still a mystery?
So again, we haven't been in humans, so it's a mystery. If you look at the statistics that have been published, if you look at the drugs that enter phase 1, 10% of them make it to approval, regulatory approval. So, drug development is a risky business, and there are a lot of unknowns. In terms of kind of what we would expect, so what immune response is when you introduce a protein, immune response is always a possibility. Part of the formulation is this is pegylated, means it's covered with polyethylene glycol. And that does a couple of things. It makes kind of a depot so that it gets a slow release from a subcutaneous, and it also kind of protects it from antibody development as well. In the animals, we haven't seen significant immune responses yet. And you might think that a human protein is even more foreign to animals than it is to humans, but at the same time, the immune systems are very different. They're very different. It's more similar in monkeys. We haven't seen that. Sometimes by itself, developing antibodies is not necessarily a completely negative thing, because sometimes you can develop antibodies, but it won't cause an immune response, or it won't decrease the impact. So this is clearly one of the big questions we have, in terms of when we're doing safety and efficacy of this is, will it work like we think it's going to do in humans, and will it be safe? And immunogenicity and immunologic response is a big thing.
Thank you. Just a quick follow-up to that. Since this is human-derived, does that give you a better chance that this may be effective in human versus a synthetic or some other derived entity?
So you might think that, and the answer is yes, but. It's always yes, but. So patients who have a different CBS than what we're giving, whether it folds differently or whatever, may recognize this as a foreign protein because their body has never seen a, quote, normal CBS. Also, it's in a different place. This is, like I said, in the blood. CBS usually is not present in the blood. So will that be another risk factor? Again, but like you said, there are other enzyme replacement therapies in which immunologic response has been a big issue. There are other enzyme replacements where it hasn't been such an issue. So time will tell.
All right, thank you.
Hi, Ben Lewis, and Gabby's not in the room, so she'll follow up with you. But I thought I'd ask a bit about the two things. First, what does the FDA define as clinical response? So, we've been collecting the data in the study around health and the side effects associated with HCU, and then general health. Are they just looking potentially for a drop in homocysteine levels as a clinical response, or are they looking for changes in some of those general health aspects as well to define that?
So in rather typical FDA way, they've been a bit vague on that. So our approach was, if we have metabolic correction, is that enough? And their answer was, you know, we need to see some clinical response. So I think part of and that may be, and so we're looking at the areas most commonly affected. You know, we're looking at kind of, you know, when Ben talked about executive functioning, that's one of the tests we do. We're looking at the DEXA scan. We're looking at, you know, the eyes, the ophthalmologic exam. You know, we're looking at some of the biomarkers. And we're trying to get a sense on quality of life. If we can demonstrate a biochemical response, a biochemical correction, and probably any of those, that would be enough. But it depends on, I think it's going to depend on how much of a biochemical correction we get. So if we take someone's level from 200 to 100, that's clearly going to be a significant biochemical response. But is that reduction going to be enough to really relate to a clinical improvement, a clinical response? So all of that is different. You know, clearly if we take someone from 200 and normalize them, I think the FDA will have a much kind of lower bar for what a clinical response is going to be than if we take them from 200 to 100. So I wish I had the answer to that question, but that's kind of what it is. That's also part of the information we're trying to get from the natural history study and stuff, because we're trying to educate the FDA, and that's why the input we got on the survey from you guys was so important today. It's like, it's so important for us to educate the FDA about, you know, what really is this disease and what is a good response.
Thanks. I might have a few follow-up questions just because.
Okay.
You know, you mentioned that the clear indicator that you've put out there is that someone's going to start around 80 and you're looking for a drop from there. That obviously relates to homocysteine level. I'm wondering if you've considered other factors and thresholds you're setting potentially around those to measure the effectiveness of the treatment. But my main second question is, so the natural history study has been going on for approximately a year or so now, I believe. You know, as a participant in that, I've gone in twice, and I will go in a third time in about a month. Is that, do you know, for patients who are eligible to roll over to the treatment within, you know, approximately 6-9 months or so, do you see that as enough baseline data to gauge the response? I mean, it's a fairly short period of time. It's a fairly small number of instances of data collection over that you know, year to year and a half.
No, you're absolutely right, and that's clearly one of the considerations we had when we kind of discussed whether we would allow patients to roll over. I think, though, this study, again, may just be 12 patients. And so if all 12 patients came from the natural history study, we'd still have the ongoing data from 22 patients. And we're still enrolling. There are some sites in the natural history study that are not going to be in this study, so that the patients at those sites will remain in the natural history study as well. It gets very complicated, and you know, it's going to get even more complicated if this study is successful and we move to a pivotal study, because then we have demonstrated that, you know, we won't have proved efficacy, but we may have proved that we have biochemical correction. And then, you know, the ethical question about why would anyone stay in the natural history study when they could go into a study where they could possibly get it. So all of these questions will kind of evolve as we come along and go, but they're fascinating questions, and they're.
Thank you.
Uh-huh.
Just one question. With regards to the exclusionary factors that have been set forth, in looking at patients with HCU that have concurrent disease processes, have those been identified with specificity as what will be exclusionary at this point in time? Is there a list? I don't see that posted at the clinicaltrials.gov.
So it's really only ongoing illnesses that we think might impact either homocysteine levels, which seem unlikely, or would really significantly impact some of the other things that we're looking to measure. But if you already have a complication from homocysteine, you know, so if you already have, you know, eye problems, or if you already have bone problems, clearly those will not be exclusionary, because what we want to do is follow those along. Our intent is to exclude as few people as possible. You know,
Okay.
we don't want to exclude people.
So would those be decisions then that are made based on your application and criteria that's set up already, or is it just based on the quantity of applicants and
So the
circumstances?
protocol will have, you know, the inclusion exclusion criteria, and the first step will be to talk to the PI and see. And if the PI has any questions, obviously we're available to talk and kind of come to the best consensus.
Okay, thank you.
We have a 5-year-old who is part of the natural history study at Children's Hospital in Boston. And I'm going to put the cart a little bit in front of the horse. If all of these things go well and the drug is proven to be effective and safe, at what age can a child be prescribed a drug like this? No, that's to be part of the study, but to actually be prescribed.
To be prescribed? So that involves a whole nother kind of interaction with the FDA or the EMA. So what we'll have to do as we develop this drug, probably, you know, if this study is successful, we will have to submit what's called in the United States a pediatric safety plan, or a PSP. And that will outline exactly how we're going to study this in children and what it's going to look like. Before we do that, we'll have to do some more animal toxicity studies. So we'll have to do what's called some juvenile studies, some juvenile toxicity studies, which are basically exactly what they are. We take small mice or rats and do toxicity in them, and we have to do some kind of reproductive toxicity in kind of the mothers. And then we'll have, you know, a plan for use in getting it into children. I think that, again, this is a very long-winded answer, but that will really depend. So in this study, you know, in the phase 1/2 study, if we're able to enroll patients 12, then even if we just have a few of those, we can take the data we get from them, especially the pharmacokinetic data, in terms of what doses we need in those kids, and we can do some computer modeling, because there's some pretty sophisticated modeling now that will predict the dose. So we can have a pediatric study plan in place that will allow us then to move into studies. And it may even be simultaneously, you know, again, how well we get our act together and what other kind of tasks we have. But it would be very good to be able to do those in conjunction with the pivotal studies. So then if and when, and again, big if, the drug is approved, we can have that in the indication label right at the time of approval. Because I think, you know, with the data, we'll be able to demonstrate to the FDA that treating children at a young age can really help prevent some of the complications. So that's a real non-answer to your question, but that's kind of the process. And, you know, our intention would be to do it as quickly as we can.
So there's no age, there's no specific age.
So,
we don't know that yet.
So we don't know that yet, but clearly, again, if we enroll kids 12 and above in this study, everything looks good, then I think that the pathway to enrolling even younger kids really will be contingent upon kind of demonstrating kind of pharmacokinetics, and we'll kind of have to pick a dose and start with them. And so they probably won't be part of the big pivotal study, but it could certainly be a kind of parallel study as well.
Okay, this is going to be our last question, and then we're going to break here for lunch, and then our panel.
Is there any point in the study where HCU patients whose total homocysteine levels are below 80 will be able to participate in the study?
Probably not this initial study, but clearly then the plan would be to expand it when we move into the pivotal study. So probably not in this initial study.
All right, wonderful. Thank you very much for answering our questions.